Okay, so the next speaker in this session is Gabriel Sturm, who will talk about his work on the association between methylation and aging and cultured fibroblasts.
Hello, everyone. It's great to be here. So my work focuses on DNA methylation age and applying it to a cultured fibroblast system. And this work has been done at Columbia University in the city of New York. Okay, so the main contextual question that we're addressing today is how do we study aging? And up till now, most aging research is done by using animal models. And this is just the few that aging studies are conducted on. However, there's a lot of evidence to say that the translation between animal models and human studies, specifically when it comes to a large, full organism process like aging, is a very difficult translation. So refining the question, again, is how do we study human aging? And for that, there are two main methods being done today, which is cross-sectional and longitudinal studies. Each study design has its own pros and cons. The pros of cross-sectional study, of course, is that they're cheaper and you can have a large cohort. However, there's a lot of environmental noise. You cannot track aging interventions, and there are many confounding variables. On the other side is the longitudinal way of studying aging, and there you get to introduce interventions and track changes over time. But the cons of that are they're extremely expensive. They take a long time to run, participants drop in and out, and usually have a very small cohort, so it's not that simple. At our lab, we wanted to think about this question a little bit differently, which is that how can we study human aging longitudinally without waiting years to study it? So applying the idea from over 50 years ago by Leonard Hayflick that cells, when taken from the body and put in culture, only divide a certain number of times before they stop dividing. And that's usually referred to as replicative senescence. And the average number of cell divisions is usually about 50, though depending on the tissue type and the individual, that's going to vary quite a lot. So that's one idea. But we wanted to take this idea one step further and say, well, if cells divide only a limited number of times in the body, perhaps you can use them as a way to track biological age by just growing them really quickly in the lab. So what normally would take years can be done in just a few months if the cells grown in culture are actually capturing the same or similar biological aging process in the body, which is a pretty bold idea. So the first thing we need to do in order to prove that idea is monitor biological age. And to do that, we are focusing on CpG methylation, right? So in the DNA of the cells, between cytosine and guanine, the cytosine can be methylated or demethylated, and there's been high-throughput methods such as the Illumina epic array, which is the data we're dealing with today, which captures 850,000 CpGs across the methylome. I would like to add that there are, of course, millions more that are not captured by this array, but this alone has already been shown to capture huge changes across human aging. So this is not work done by us, but work done by Steve Horvath, who's a collaborator on this paper, on this work, where he takes those arrays, those massive amounts of methylation data, and then runs an elastic net regression on that, which is like a lasso-esque regularization that chooses the best, in this case, CpG sites that can predict age, and then outputs a clock or an age predictor that associates the actual chronological age of a person with the predicted biological age based off specific, maybe 300 to 500 CpG methylation sites that are most age-associated. So this has been a very, very powerful clock that was designed, and there are many clocks, but these clocks have been able to predict many aging phenotypes, including Alzheimer's and cancer, and morbidity and mortality. All these things have been associated with the predictions outputted by DNA methylation clocks. And if you want to read more about these types of predictions, you can see Steve's paper. So these clocks capture aging in the body. So now we're going to design an experiment where we'll take those clocks and test them on cells grown in culture in the laboratory. So to do that, in this case, it's just a small subset of data where we have a male individual, a healthy 29-year-old. We take fibroblasts from the upper arm, put them in culture, as seen here, and then we can give them different age-modifying treatments. In this pilot experiment, we just give them normal glucose and diabetic levels of glucose on the cells. And then here's the main part of the experiment. We grow them for about 4 months and just continually make sure that the cells keep on dividing, so to proliferate them as much as possible. And as they're dividing, we take cells from them and measure DNA methylation. So this data is going to be about 12 time points of normal glucose and 14 time points of diabetic glucose. And for those who are unfamiliar with the laboratory, this is a biological hood with a microscope and cell counter, which is all that it takes to run this type of experiment. It's pretty simple. Okay, so then putting this all together, we have data taken from actual people that was able to predict age, and now we're growing cells in culture and tracking their aging in a much more condensed fashion. So that usually humans take years. Here we're doing it in just a couple hundred days. And there'll be 3 main findings of this work, is that there's a conservation of the methylation patterns, the aging methylation patterns, at a global level, at a gene level, at a CpG level. So 3 reducing levels of observation all show a conserved and accelerated aging process. Okay, so here's the classical growth curve of our cells, where you can see that the cells grow at a pretty continuous rate before they reach a point where they plateau and they stop dividing. So on the y-axis is the number of divisions that the cells have gone through, and the x-axis here is time, the days that the cells grown. So for about 110 days, the cells were growing before they started to slow down. And if you want to know what the cells look like, these are fibroblasts, they form kind of skin layers or concentric circles, but then as they get older, they lose that ability and they start forming these weird senescent-like morphologies. Okay, so here's again the main question. We have these really cool clocks that can predict age in the body. What happens when you take the methylation patterns from cells, and what do they output? So here is the blue line is the normal glucose, right, the control group here. And you can see that it predicts the person to be 29 years old, or about that age, which is really good. And then as you divide the cells, the age increases. And this is really rapid. So this is just 120 days, and you go from 30 years all the way to 55 years. So that's 25 years of aging in just 120 days. And if you're curious, the diabetic glucose, which was supposed to be an age accelerant, does in fact, in terms of the elevation of the glucose line, is a 3.5 years age acceleration, which is what we would expect. And then here's the key point, is that the slope of this line is 60 times faster than the in vivo rate. The in vivo rate, of course, is 1 biological year per chronological year, but the in vitro rate, the culture rate, is 60 times that. So that's the key finding at the global level. But moving on to the gene level. Now, to study the genes, we wanted to do is compare them to actual data taken from people. So to do that, we collaborated with the Karolinska Institute, and they have the Swedish Twin Registry, where they have twins measured longitudinally across up to 15 years, I believe. In this case, it's 385 participants taken on whole blood. Very similar design to our experiment, but just actual people over time. And in the beginning, we focused in on the ELOV-L2 gene, so just one gene that happens to be the most age-associated gene in the body in terms of methylation. So various papers have found this CG168, 67, 657 to be the most age-associated gene, and most age-associated CpG site. So looking at just that one site, what you're seeing here on the y-axis is the level of methylation, and on the x-axis is kind of the methylation sites across the gene, so going from upstream to downstream on the DNA. And the coloring here is the level, the age of the individual or population. So on the top is the cultured people, sorry, the people from, it's the actual people, and the bottom is the culture. So here's that site that's most age-associated, and you can see that there's a very similar pattern across the entire gene, and specifically at that site, there's quite a robust increase in methylation, hypermethylation across age in both the body and the person. And just to show that more clearly here, there's a 21% increase in the body and a 35% increase in culture. And what's great about having cells now is we can also measure really accurately the RNA transcripts, the level of expression of those genes, and you see an 80% reduction in gene expression of ELOV-L2. So we did, well, I'm not showing here this today, but we did scale this up to quite a few more genes, and we found about 75% of age-related genes do in fact have similar methylation patterns, and 50% of their methylation sites are age-associated. But I didn't want to get to so much of those details, just because I want to get into the third point, which is since we have a high temporal resolution system here, meaning we can collect points almost as often as we'd like when we collect this data, as opposed to people where you can only bring them into the clinic every so often. So since with that high temporal resolution, we can see patterns of methylation a lot clearer. So here, this is a principal component analysis, where here's the youngest sample and here's the oldest sample, and you have this V-shaped effect across the entire methylome, which if you break it down is principal component 1 is linear, and principal component 2 is kind of this inverted U shape. So we wanted to see a lot of these clocks are built on kind of linear changes over time, and we wanted to explore perhaps the nonlinear changes that happen across aging. So to do that, we take the top 1,000 most changed methylation sites across age, and then we group those using generalized additive modeling into different degrees of freedom. So that should be different levels of complexity and changes across time. And then group those into ones that are hypermethylated, those are increasing with age, and hypomethylated ones that decrease with age. And that's the groupings you can see over here. And then we graph the top 20 of those sites. Okay, so here, the y-axis here is the level of methylation from 0 to 1, or 0% to 100%. And the x-axis here is time. And you can see that some sites are flat with time, some sites are linear with time up and down, some sites are quadratic, and other sites cubic. And I believe this is the first time that it's been reported about a methylation site being having these relatively complex changes across age. And that's going all the way up to like 47% decrease in methylation per month, which is a very rapid decrease in methylation. So this is all very exciting. And to put it together again, what we're showing is that there is in fact a conserved methylation pattern at the clock level, the gene level, and the single CpG level that is both not only conserved but accelerated. Now, of course, I want to say that this is kind of a proof of concept work. It's only done on one individual, and we've already put in the work to show this with many other individuals included in the study. So hopefully that data will be soon to validate this. And what makes me really excited about this work is it leads to a situation in which we can potentially study human aging longitudinally at an extremely accelerated rate, as I said. And what that could do is maybe you can design high-throughput screening platforms for aging targets. So quickly assess whether or not an aging target is actually affecting the aging process is a very preliminary assessment by looking again at the rate of aging, which in this case is 60 times faster, and we can just see if that slope changes more or less to know if that target is actually slowing or speeding up the aging process. So that's my talk, if you have any questions. This is the acknowledgments of all the people that are involved. It's quite a team effort from many, many institutions, and yeah, thank you.
We have time for questions.
Nice talk, thanks. Quick question on, does the methylation age predict the time to senescence of cells?
Yeah.
And if you control for the biological age, does it still predict the time to senescence of cells?
Mm-hmm. We haven't looked into this, but I will say that different clocks kind of plateau during senescence, meaning assuming that cells are no longer increasing in age when they reach senescence, while other clocks, their sites continue to increase even through senescence. So it really depends on which methylation clock you use.
But there would be different times to senescence for different individuals. And is that,
yes.
Is that interval predicted?
Yeah,
by
we would have to look into
age?
that, but
Okay.
I absolutely will.
Okay.
Yeah.
So, so what is it about the, if you know,
Mm-hmm.
the cell culture that makes aging happen faster? And if you do know,
Yeah.
what can you do to make it happen even faster yet than the 60-fold speed up?
Great question. We're certainly, it's ongoing experiments that we kind of have partial answers for. One idea is you would think that just the fact that the cells are dividing so fast, that's why they're aging so fast. But you have to realize that when you're taking cells out of the body, there's a lot of environmental changes that are happening. One example of that is oxygen levels in the body is 3%, in the environment is 21%. That could also be a contributor to the age acceleration. So we're kind of tinkering with those different variables and seeing if it brings it back to the in vivo rate.
Yeah, for the genes with the acceleration pattern, do you see any molecular features that they share? Any gene ontology enrichment? Did you look into that?
We tried it. It didn't work. So finding two groups of genes that might all show similar aging patterns. Yeah, we couldn't find any specific groupings. Yeah,
Okay, thank you.
of course.
Hi. So we work quite a lot with fibroblasts as well in the University of Manchester to study aging. So I was just wondering, because we are planning to move into the actual 3D cultures, would you consider actually trying something like that out as well? 3D cultures, because there is a big difference between fibroblasts in the 2D culture and 3D culture. And another question that I had is, what would you expect to see if you took fibroblasts instead of from 20-year-old individual instead from 60-year-old?
Yeah. So both questions were, well, certainly the second question, we have kind of an array of fibroblasts from different ages, and we're going to see if the rate of aging is different between them. And regarding your first question, I think it'd be really cool to do 3D cell culture. We're not quite set up for that at the moment, but I think it'd be really exciting to try.
Okay.
Yeah.
So when Steve Horvath showed his initial clock CPGs, they didn't exhibit any consistent pattern in which you could sort of see a decreasing over time. It seemed to be more stochastic kind of behavior. But you're telling us that there's actually a difference in your fibroblasts. You're actually seeing some consistent patterns of CPGs over time changing. So can you sort of explain the difference with Steve Horvath's initial results?
Yeah, I mean, I'm not familiar with Steve's results in that case. But yeah, I guess it's very possible that since it's a culture condition, everything is super stable, and you know exactly what you're giving them, and tracking them over time is just almost just like letting them divide, and there's very few other things changing. That might allow kind of the signal to come through that wouldn't otherwise come in Steve's data, which is actual people. That'd be my guess, but I don't know.
All right, thank you.
Let me also ask a question. I wonder about the tissue specificity of these aging models. So you use fibroblasts, which I think is an obvious model for culturing cells. But I could imagine that the aging clocks, the models would differ quite a bit if you look at different tissues. Have you thought about that? Or
Absolutely. I'm curious if you just take non-proliferative cells, neurons, whatever it may be, will they show an increase in demethylation agent culture? But there are plenty of other proliferative cells in the body beyond fibroblasts that would be great to test out. I guess I said this is just the beginning of where this can go. So yeah, great question.
All right. Okay. Thank
Thank
you.
you.